Good morning at home. Just 3 people. Anyway. Okay. So yesterday we just stopped here in the describing the smooth muscle. Okay, and we already know that the classification of the smooth muscle is a bit complicated because it depends from the um, location. So we have vascular, gastrointestinal, urinary, respiratory, reproductive, and ocular smooth muscle. Then we classify the muscle by the contraction pattern, and we have the phasic smooth muscles and tonic smooth muscles. We will see right now the type of the contraction. And then by their communication with neighboring cells. So because the smooth muscle can work as a single unit motor or multi-unit motor smooth muscle. Um, Everything is okay at home?
We cannot see the slides.
Ah, okay. Sorry. They are right. What about now?
Yes, we can. Thank you.
Okay. Okay. So let's see the point two. Okay, we described yesterday about the location. What about the contraction pattern? Here you can appreciate the 4 type of contraction pattern in smooth muscle. Let's analyze considering the contraction pattern. And as you can see from the graph, the smooth muscles is quite different from the striated muscle that we analyzed last week, okay? And also it's different the way in which they develop the tension, the force, the contraction force, okay? In particular, the smooth muscle cells can be classified by whether it alternates the contraction and the relaxation states, or if it is continuously contracted. What do you mean? What does I mean? Let's analyze the phase, the panel A and the panel C. Okay, in the panel A you can see the smooth muscle that is usually relaxed, like the esophagus. A stimulus can start a contraction in smooth muscle cell, in the phasic smooth muscle cell, but this muscle is usually relaxed. By the time the muscle goes in the contraction state and then in the relaxation state here. What is the difference between the phasic smooth muscle and a tonic smooth muscle? A muscle that is tonically contracted, that is usually contracted, continuously contracted. You can see here the example in C. A sphincter that relaxes to allow the material to pass, to move. And you can appreciate this point here. In the tonic smooth muscle, the muscle is always usually contract. Here is the muscle is contract. Here is relaxed. Okay, so in the tonic smooth muscle that is usually contract, a stimuli can relax the muscle. The sphincter, for example, a sphincter we know from human anatomy that we have muscles that are called sphincter, that goes to a relaxed state, and the material can move through the organ because the smooth muscle cells are relaxed. And then the muscle goes again in its contraction state, returning to the contraction force at the beginning, similar to the beginning. So this is completely different, because in the tonic smooth muscle, the contraction is, the muscle is usually contracted. In the phasic smooth muscle, the muscle is usually relaxed. In panel B and panel D, we can see other type of phasic and tonic muscles. In panel B, for example, we have the muscle that is present in the intestine that is characterized by the relaxed state at the beginning, and this muscle can undergo to periodic contraction and relaxation states typical for the intestine or esophagus or stomach. Okay, this is a phasic smooth muscle. Differently again, a tonic smooth muscle, like this one, this is a vascular smooth muscle that is usually tonically contract. You can see here, this muscle is contract, continuously contract. These muscles are called tonic smooth muscle. And they can be relaxed, but they return to the contraction, to the initial contraction. So these are really different muscles, always smooth muscles, but phasic and tonic smooth muscles. And here we have analyzed the group, the big group of smooth muscles by the contraction pattern, tonic and phasic. Now, what about their communication with neighboring cells? Yeah. Again, we have asked the example of the small intestine and the eye. In this case, we have a single unit smooth muscle cell. Why single unit smooth muscle cell? It are also named visceral smooth muscle. Visceral smooth muscle. Why? Because forms all the wall all around the viscera. But the most important point is that all the cells are connected by the gap junction here. What is the role of the gap junction and how the gap junction are built in the smooth muscle cell? We already have studied the gap junction. Do you remember where? Where did we analyze the gap junctions? In which part of the program that we have already studied? In the synapses, for example, in the electrical synapses. And we already analyzed the gap junction in terms of the structural component of the gap junction, such as the connexin that forms the connection, okay? We studied that at the beginning of the nervous tissue, okay? So remember it, because we can talk about gap junction also here, totally in the nervous system. But in this case, okay, what is the meaning, the physiological of gap junction here? Or the smooth melt. Nature has built up so many gap junctions here in the smooth muscle cells. And the physiological role is because the cell can contract as a single unit because all the cells are connected one to each other. So if the neurotransmitter is released here, look at the green ball here. If the neurotransmitter is released here, smooth muscle cells here on top of the intestine can be contracted. So the more far away smooth muscle cell here can be contracted. Why? Because they are connected by gap junctions, and current inside of the cell can reach all the cell around the muscle, the smooth muscle. This structure here, the autonomic neuron varicosity, because you remember perfectly from human anatomy that we have the efferent part of the nervous system, the peripheral part of the nervous system that is called sympathetic and parasympathetic component that innervate the muscles, the smooth muscles, and the autonomic neuron varicosity release the neurotransmitter, and the release of the neurotransmitter can produce the muscle contraction. How is the name of the junction between the motor neuron and the skeletal muscle? Do you remember what is the name of the junction between the motor neuron and the skeletal muscle? Is called the neuromuscular junction between a motor neuron and a skeletal muscle. Here we have more or less the same physiological structure, but the components are different. The autonomic neuron varicosity from the autonomic part of the nervous system that controls all the involuntary movement of our body, and the release of neurotransmitter can produce the contraction of the muscle. The second component, completely different, okay, and it's showed here in panel B, shows the multi-unit smooth muscle cells, where the cells are not linked together, and in this context are not electrically connected. The gap junction are not present. And they must be stimulated independently in order to contract. And you can see the varicosity here in the deepest part of the muscle, of the smooth muscle. And I would like to you to notice that each individual muscle cell is closely associated with a varicosity, with a axon terminal with the varicosity. So this cell here can be contracted only because the neurotransmitter is released here. Okay? They contract independently. They must be stimulated independently. And as I told you before, I don't want to spend time with this slide because you should already know the structure of the somatic motor pathway because we already analyzed it, and the autonomic pathway with the parasympathetic and sympathetic pathway. Okay, anatomically, we know from human anatomy how they are constructed. Okay, the motor neuron that voluntarily we can contract the skeletal muscle cells by the release of acetylcholine on nicotinic receptor, so the muscle can be contracted. We know everything about this portion now. What about the autonomic pathway? The pathway that connect the sympathetic and parasympathetic nervous system to the target cells that are smooth muscle cells, but also cardiac muscle cell, some endocrine and exocrine glands, adipose tissues. All of them are autonomic targets. We cannot control our intestine, okay? We cannot control the cardiac muscle. So they are autonomic targets. They have to maintain the homeostasis. A lot of roles of these targets is just to maintain the homeostasis. And the next slides, you will analyze the autonomic pathway with Professor Marsenzio. But we already, sorry, we already know about how the neurons can talk to each other. Here you have a pre-ganglion neuron. We have here a ganglion and a postsynaptic, post-ganglionic neuron. What is this? This is a chemical synapses, and the signal goes from the central nervous system to the ganglion. The signal is transduced in a chemical signal, and again the chemical signal is again transduced to a electrical signal, and then chemical signal directly to the smooth muscle cells. And the same with the sympathetic pathway. You probably know again from human anatomy that the parasympathetic and sympathetic portion, they have a double control. One is excitatory and one is inhibitory, okay, in general. but you will study with Professor Masenzio deeply. So I want just to use the next 2 or 3 slides that are important because they shows you from a cellular point of view the differences between the efferent neuromuscular junction and the somatic neuromuscular junction. They are completely different. If I ask you during the exam, tell me something about the somatic motor pathway and the autonomic motor pathway, you can talk about a lot of things. Not only this is voluntary, this is involuntary, but also from a physiological, cellular point of view. You have here the acetylcholine, you have here the acetylcholine and the acetylcholine in the ganglion and the noradrenaline in the structure, in the tissue, in the target, and here the acetylcholine in the target. But how? Is the same this structure? Compare this structure? No. The two structures are completely different. Again, the autonomic synapses, okay, contain the autonomic axon, postganglionic axon from the autonomic neuron that forms this type of varicosity all around the muscle. And why? We have described it before, because if the smooth muscle is a multi-type of muscle, so the cells are not connected, every cell can be contracted only if the neurotransmitter reach the cell. And how is possible? It's possible because the autonomic varicosity that contains the neurotransmitter, obviously, okay, release the neurotransmitter on top of the muscle. And every cell that contain the receptor can be contracted. Let's see an example here, okay? The norepinephrine, okay, noradrenaline, is released by the sympathetic nerve effector junction. This is a neuron. Okay, I'm just showing you this type of synapses, okay, from the autonomic pathway, because we already described it, this one, okay? So the autonomic contains the parasympathetic and the sympathetic pathway. One is inhibitors, in general, one is inhibitory and the other one is excitatory. The sympathetic in general is excitatory, the parasympathetic is inhibitory. From a structural point of view, they have the preganglionic neuron, this one and this one, preganglionic neuron, okay? They start from the spinal cord, and then they have a ganglion outside of the body, and then the postganglionic neuron from the ganglion to the autonomic targets, okay? And they release in the ganglion acetylcholine, always acetylcholine on nicotinic receptor. They are the same, either parasympathetic or sympathetic pathway. The difference is in the autonomic target. The parasympathetic pathways contains the acetylcholine as a neurotransmitter on muscarinic receptor, while the sympathetic pathway contains the noradrenaline on noradrenergic receptors. Have you studied the autonomic pathway in human anatomy? Yeah, okay. So, let's see this example. Norepinephrine, okay, is released here in the neuroeffector junction. Neuroeffector junction. How is released here the norepinephrine? The mechanism is always the same. The action potential goes from the postsynaptic neuron all along the neuron and move until reach the varicosity. When the action potential reaches the varicosity, what happens? A voltage-gated calcium channel open. Why? Because it's voltage-gated and because the action potential depolarizes the neuron. A calcium ions move inside of the cell, depolarizes again and more the neuron, but the most important thing is that the calcium entry triggers the exocytosis of synaptic vesicles, in this case containing the neurotransmitter norepinephrine. Norepinephrine binds to the adrenergic receptor, that is a G-coupled receptor sometimes. And what happened in the target cell? For example, can be the result, can be the response, can be a contraction of the smooth muscle cell. And what about the neurotransmitter? The norepinephrine is removed by diffusion from the synaptic left, or can be also transported, removed from the synapses by uptake into the synaptic vesicle for a new release, or can be also metabolized by the monoamine oxidase. Is the enzyme that metabolize the norepinephrine. Which is the name of the enzyme that metabolizes the acetylcholine neurotransmitter? Do you remember?
Acetylcholine esterase.
Exactly, perfect. Acetylcholinesterase. Okay? Thank you. So, the neurotransmitter needs to be metabolized to stop the activity of the neurotransmitter. So, this is just an example, okay? An example that is for the sympathetic pathway, because norepinephrine is released here and the response is to norepinephrine. In the parasympathetic pathway, the neurotransmitter is acetylcholine. The postganglionic neurotransmitter is the acetylcholine on top of muscarinic receptors. And again, remember, when we studied the receptor, the response of the target depends on the type of the receptor that is present. Here you can see the alpha receptor, beta 1 receptor, beta 2 receptor. There are other type of receptors also that are pharmacologically modulated. You will study it soon. But the response of the autonomic target depends only to the receptor, because the norepinephrine is able to bind to all of them. And obviously the response depends on which type of receptor is present. And the example that we did, this one, is just an example of what happened in the postganglionic cell and the target cell. For example, the muscle cell. So we didn't study it yet, but what could be the response of a smooth muscle cell to a neurotransmitter? To contract. And what do you think that is able the norepinephrine to do on the adrenergic receptor? What is the tricky point here? What could be the response from a mechanicistic point of view? Contraction, it's okay, but what is important for the contraction? What is important for the contraction in the skeletal muscle? Calcium. So here is the same. Here, calcium is important. In ATP, obviously, okay? But calcium is the tricky point, the important ions that we need to perform the contraction. And there are differences between skeletal muscle and smooth muscle in terms of where the calcium comes from. But in both muscles, and also in the cardiac muscle, the calcium is extremely important. But let's see, since we have already described the type of the classification of the smooth muscles, let's see the organization of the smooth muscle, okay? Because also in this case we have actin. You look at the laser here, actin in red and myosin here in purple. Okay, so we have the same filaments that produce the force of contraction. The force of contraction is created by the filaments of actin and myosin that cross bridges and allow the sliding of the filaments in the same way that produce the contraction in the skeletal muscle. The contraction also in this case is initiated by an increase of calcium ions. How is the concentration of calcium inside the cell? How is the concentration of calcium outside the cell? Okay, remember. Both these two characteristics of the skeletal muscle are present also here in the smooth muscle and in the cardiac muscle too. It's the same. But there are numerous other differences because smooth muscle cells function very differently from the skeletal muscle. Let's see some of these anatomical and morphological differences that again reflects functional differences. For example, is present the sarcomeric organization in the skeletal muscle, in the smooth muscle? No. The smooth muscle cell lacks the typical band organization. If you remember, one slide at the beginning shows you that it's completely disorganized, okay, or at least not well organized, such as the skeletal muscle. In this slide, you can appreciate here the structure is formed by actin, myosin, cross bridges here, the sarcoplasmatic reticulum is still present but is reduced. There are these portions that are called this one dense corp dense, the pink, the purple square here, dense body. What is a dense body? What do you remember? What can be parallelized by the corp dense in the skeletal muscle? What do you think that the dense body is in the skeletal muscle?
Like you have the vessel inside, maybe, like artificial vessels.
No, the sarcomere is delimited by what? Do you remember the sarcomere is delimited by two band
I can't remember the right name, but
lines, the Z lines, okay, in this structure. Okay, and from a Z line to the other Z line, the actin start to move inside of the sarcomere. Okay, the dense body is more or less the same from a structural point of view. From one dense body and the other, the actin move in the center of the muscle. And what is inside of the myosin, the purple myosin, and the myosin filaments contains the same structure. The heads that are able to attach to the actin and to produce the power stroke. In the same way, the myosin is a little bit different. I will tell you in a moment, but the filaments is more or less the same. Then the actin is attached to the dense body, and each dense body is connected by filaments. So the structure is disorganized compared to the skeletal muscle, and when this dense body, when this actin and myosin contract, one dense body goes close to the other dense body. So, why? Because the myosin filaments move in the sliding way, in the sliding mechanism, in the actin filaments, on top of the actin filaments. So, in this case, the two proteins that are responsible for the power stroke, again, actin and myosin, produce the same type of contraction. There are a few differences. For example, actin is more present, plentiful, compared to myosin. 10, 15 filaments of actin compared to 1 filaments of myosin, more or less. The actin is associated also in this case with the tropomyosin, but smooth muscle cells lacks the troponin. Do you remember the role of troponin during the contraction of the skeletal muscle?
Yeah, it's the calcium.
Exactly. Binds to calcium and it's useful for, it's important for the contraction. So in the smooth muscle cell, since lacks troponin, there should be another mechanism, a physiological mechanism, and we will see it, okay? But this is a structural and morphological difference, lack of troponin. The actin bound directly to the cytoskeleton in the dense body, okay? In the blue, in the purple circle here. In the muscle cell, in the smooth muscle cell, myosin filaments are longer, and the entire surface of filaments is covered with myosin head. So this myosin head, here and here, can move all along the actin filaments. Can you imagine the two dense body? They can be really attached when the muscle is contract one to the other, because the myosin filaments contain the head on top of the entire surface of the filaments. Another difference is that there is an extensive cytoskeleton, not only here in the intermediate filaments, but also because inside of the cell and outside of the cell, let's see here the connective tissue. What is the role of connective tissue? That is not the gap junction, the connective tissue. It's just they are just protein that connect one smooth muscle cell to the other. So when one muscle cell is contract, the other one is stretched and goes under the contraction at the same time. As I told you, the sarcoplasmatic reticulum distribution varies and is not really well organized. not also present. The T-tubules are not present. You can see it, they are not present here, T-tubules. But obviously the smooth muscle cells are smaller compared to the skeletal muscle. In some cases, there are structures that are called caveoli here, caveoli that are stores for calcium ions. What else? These protein fibers, we already described the connective tissue, the protein fiber that in the extracellular matrix here help tie the smooth muscle cell together and transfer the force from a contracting cell to the other. So we have a lot of differences between skeletal muscle and smooth muscle organization. But what about, and this is the same, always the same resume slide, but what about the single muscle contraction in the skeletal muscle and in the smooth muscle? Look at the graph. Here is the tension, the force, the contraction, and here is the time. Obviously, the difference is enormous. You remember the tension of the skeletal muscle? We said that could be from 10 to 100 milliseconds. In the smooth muscle, it's really a big amount of time, like 5 seconds. In physiology, 5 seconds are a big amount of time. So, smooth muscle are the slowest to contract and to relax. Smooth contract and relax much more slowly compared to the skeletal and compared also to the cardiac muscle. If you pay attention again to this graph, there is also another characteristic of the smooth muscle cell that is really appreciable from the graph, that the force developed from the muscle cell is not so rapid compared to the tension originated by the skeletal muscle. You can see here from the from this part of the curve. You can reach the top of the contraction in 2 milliseconds. In the skeletal muscle, you reach the top of the contraction in very low amount of time. So the smooth muscle cell can develop force very slowly, and slowly can relax also. And this characteristic is due to the ATPases activity. The ATPases activity of the myosin in the skeletal muscle is slower compared to the skeletal muscle. They have the ability to slow down their myosin ATPase activity so that the cross bridges cycles slowly, And also they can maintain their force for a long time. As a result of this behavior, their use of the ATP is very low compared to the striated muscle. It's slow, so they use less amount of ATP. It's quite obvious. In fact, the smooth muscle cells has less amount of mitochondria and relies more in glycolysis for the ATP production. Physiologically, the smooth muscle cell, for this property, can maintain contraction for extended periods without fatiguing. Let's think to the bladder and constantly maintain the contraction even if it's continuously filled, loading with urine, okay? Or let's think in the tonic muscle that we analyzed before. They spend less amount of ATP to maintain the contraction. Why? Because the ATPase activity is low. So, you want to stop for 5 minutes? Okay, let's have a break for 5 minutes. We have the next lesson on Friday and then Monday, then we finish. I finish my module, so I know that you are extremely sad about that, but the best always finished. Yes, 11th of April. Then you will start the lessons with Professor Masenzio and also the class of laboratory, online laboratory,
Okay.
with Dottor Formaggio.
Perché ho visto nel schedule anche dopo abbiamo ancora corso con lei, quindi no, questi...
Con me dopo sì, ci sono anch'io, però le lezioni, i laboratori le facciamo io e il professor, e il dottor Formaggio. No, no, tranquilla, mi vedrete ancora. Okay, so we have just analyzed few information on the structure of the structure and function of smooth muscle cells compared to the skeletal muscle cell. Again, another question could be, tell me the differences, the molecular, structural, and functional differences between a skeletal muscle and a smooth muscle. Okay, for example. And in the next two slides, we can see other structural and functional differences comparing the two muscles. For example, you can see here smooth muscle must operate over a range of length. Okay, again, we already analyzed it, but again, think on the bladder that contain smooth muscle cell all around the bladder into the wall, okay, and is going to fill with urine, but not all urine at the same time. Is going to fill the urine in the bladder in a time-dependent way. Okay, so the smooth muscle cell here in the bladder needs to be contract, but in a over of different ranges of length, dependently on how much urine is inside of the bladder. Muscle like this has to operate over a range of length. In contrast, in the skeletal muscle, the contraction is immediately the best contraction that you can perform, while the smooth muscle in the bladder that contains the urine needs to work to operate over a range of length. It depends from the urine that is produced. Okay. Another interesting difference between skeletal and smooth muscle cell is that within an organ, the layers may run in several directions. We already have analyzed, I think, this point, okay? Again, another example, think in the intestine, okay? And remember that the structure from the human anatomy that you have studied last year. The intestine is an hollow organ and have to move the food in one direction. But it's just not only a moving of the food from the mouth to the ano. There are a lot of different functions all around the tube. And the gastrointestinal tract contains different type of muscle. Do you remember the type of muscle that we have in the gastrointestinal tract? Smooth muscle, but which type?
Several single units of muscle.
No, but which kind of muscle? Which type of smooth muscle? We have the circular smooth muscle and longitudinal smooth muscle, okay? And they are different from a structural point of view. They are smooth muscle, but in one way they are circular and they perform this kind of movement, okay? And in the other way are longitudinal. They contract in this way, while the circular contract in this way, okay? So the intestine contains the circular and the perpendicular longitudinal cell, okay, smooth muscle cell. And which is the role of this smooth muscle? Why? Nature has built an intestine with two types of smooth muscles. Why?
To make food, to move food.
Okay, to move food, yes, but to move food is just necessary one of these muscles, the one that works like this, like a sausage. If I want to empty a sausage, just I push it, the circular muscle like this, okay, and the food moves. So which is the role of the other, the longitudinal muscle?
Can you reach to support the circular ones?
To support in which way?
Like guarantee the movement.
But the movement, if I move, this is the longitudinal muscle, no? And this is the circular. Doesn't.
Yeah.
But the longitudinal muscle has one important role. Which could be the important role? For this, with this one, with the circular, I move the food from the oral movement, no? From the mouth to the anus, with this one. And with this one? What do you think?
To mix the food.
Exactly. No, it seems you're smiling, but the real point is this one. You mix the food because inside of our gastrointestinal tract there are enzymes that needs to encounter the perfect site where they have to perform their function. So the food is mixed. So move from one direction to the other direction, and this move the food. Okay, so the food can be metabolized by different enzymes, by the contraction of these two different layers, smooth muscle layers. Okay, so we already have discussed about the point 3, 4, and 5. Okay, in the red color, and the point 6 we have already discussed, but I mean, just remember, if you remember the slide, I will show you. Okay, or this one. Okay, this one. Or this one. Okay. Point 6 says that the smooth muscle cell have small spindle-shaped fusiform cells with a single nucleus. Okay, completely different compared to the skeletal muscle, for example. And point 7. The contractile fibers are not arranged in sarcomeres. We have already seen this point. Okay. Point 8. Contraction in smooth muscle cells may be initiated by electrical or chemical or both. This is completely different from the skeletal muscle. The skeletal muscle can be contracted by just one mechanism, and is the neuromuscular junction, acetylcholine and choline receptor, nicotinic receptor. The smooth muscle cell can be contracted by an electrical, chemical signal, or or sometimes the smooth muscle cell can be contracted by just their self, without any type of stimuli, we will say. The smooth muscle cell is controlled by the autonomic nervous system. Smooth muscle cell lacks specialized receptor regions, means that there is not a neuromuscular junction, okay, but the neuroeffectors, as we said before, and point 11 and point 12, the most important for the contraction. The calcium for contraction comes from the extracellular fluid as well as from the sarcoplasmatic reticulum. Where the calcium comes from in the skeletal muscle? Sarcoplasmic reticulum, only sarcoplasmatic reticulum. Here, the calcium can be extracellular or intracellular calcium. Good. And the calcium signal initiates a cascade that ends with the phosphorylation of myosin light chain and activation of myosin ATPase. In skeletal muscle is very rapid. Do you remember the action potential? The T-tubules go inside of the muscle, the ryanodine receptor, the DHP receptor, the calcium goes out from the reticulum. Here, the calcium initiates a cascade, so a little bit slower, that ends with a phosphorylation of myosin light chain and activation of myosin ATPase. And then contraction. Let's see the complete process here in this slide. Process of smooth muscle contraction and smooth muscle relaxation. As we said a few seconds ago, okay, calcium can be extracellular calcium or intracellular calcium or both. The complete process of contraction and relaxation from a structural point of view, the myosin and the actin just slides one on top of the other, like in the skeletal muscle. But the players here are a little bit different. Let's analyze the step one by one, okay? The increase here of cytoplasmic to cytosolic calcium initiate the contraction. But pay attention, the calcium ions can come from the outside or the inside of the reticulum, of the sarcoplasmic reticulum, even if it is not so organized or so present compared to the skeletal muscle. Remember, again, calcium concentration outside, calcium concentration in the cytosolic compartment, calcium concentration in the sarcoplasmatic reticulum. What happens when the calcium inside of the cell increases? Calcium binds to a protein that is called calmodulin here, and the calmodulin is a protein that is calcium dependent, that is present inside of the cell, that contain obviously calcium binding site. When the calcium binds to the calmodulin, it activates a myosin light chain kinase. Kinase is a phosphorylation of something, okay? So in this case, the calcium calmodulin binding site is the first step that initiates the cascade that I told you before, the cascade that is result in the phosphorylation of the myosin light chain. The complex calcium calmodulin activate an enzyme named myosin light chain kinase. And what is the role of the myosin light chain kinase? The myosin light chain kinase phosphorylates the myosin light chain here, and this phosphorylation enhances the myosin ATPase activity. The myosin ATPase activity is increased. Obviously, the power stroke can be determined, and the cartridges lie along the actin, create the muscle tension. Only when the calcium increase inside the cell can activate the complex calmodulin activates the myosin light kinase, an enzyme. The myosin light chain kinase phosphorylates chains in the myosin head and increases the myosin activity. And the phosphorylation happens here. The muscle phosphorylation here increase the myosin ATPase, the ATPase increase the phosphorylation coming from myosin light chain kinase. So at the end, the contraction can take place, okay? Look at here. You remember the 45 degrees angle and the 19 degrees angle in the skeletal muscle, okay? Here I am in the 45, okay, 45. If I'm going to 90, now I have a lot of energy in order to move the actin, okay? And here is the same. When the ATP is present and the phosphorylation of the myosin light chain kinase happen, the myosin head has the 90-degree angle. So now the myosin head is able to power stroke the actin and move the two filaments and increase the muscle tension. Remember two things: the calcium increase can be outside or inside of the cell, and the base of the contraction in the smooth muscle cell, at the base, I will show you, I put here the same structure that we have seen in the skeletal muscle cell, just to show you that here is the light. Okay, so at the base of the myosin head, there is this small regulatory protein called myosin light chain. The phosphorylation and the dephosphorylation of the myosin light chain here, okay, can produce, can control the muscle contraction and relaxation. Only when the calcium enters to the cell and combine to the calmodulin, this process is activated. Calcium calmodulin activate the myosin light chain kinase that phosphorylate the light chain and produce the power stroke. This is the contraction. What about the relaxation? If calcium pumps, such as the PMCA, the SERCA1 that we already know perfectly, or active transport, move the calcium outside of the cell, the free calcium ions in the cell, in the cytosol, decreases. So the complex calcium calmodulin decrease the activity of the MLCK. So the light chain is not phosphorylated and the contraction is inhibited. But another important point here, you can see in the point 8, the myosin phosphatase, myosin light chain phosphatase, removes the phosphate from the myosin light chain, which decreases the myosin ATPases activity. It's an enzyme, so they have also the myosin phosphatase activity. In this case, is a phosphorylation. In this case, is a dephosphorylation. And when the myosin phosphatase activity is active, the muscle decrease the muscle tension. When the less myosin ATPase activity is produced because the MLCP is activated, the result is a decrease in muscle tension. In this right part, in the relaxation process, the dephosphorylation of the myosin light chain by the enzyme MLCP, the myosin light chain phosphatase, decrease the myosin ATPase activity. So the dephosphorylation, the dephosphorylated myosin, obviously do not produce immediately, like in the skeletal muscle, a relaxation. Can be retained, attached to the actin for a period of time. This is called a latch state, okay, a block state in the smooth muscle cell. This is a typical state, latch state, when the muscle can remain contract for a longer period of time. But in any case, when the myosin phosphatase activity is present, the muscle decrease, the smooth muscle cell decrease the tension. Okay, so two different activity, enzyme activity that are linked to the calcium increase in the cell. Calcium calmodulin activate a myosin light chain kinase that phosphorylate the light chain in myosin heads and increases the myosin ATPases activity. If the myosin ATPases activity is increased, the power stroke can be increased, and in turn the muscle tension can be increased. In the other case, the myosin phosphatase MLCP, myosin light chain phosphatase activity remove the phosphate. So the process is the opposite. If I remove from here the phosphate, the ATPase activity decrease and the muscle decrease the tension. It's okay? Okay. So you already know now that it's important not only the amount of the kinase activity compare is not the amount of the total kinase activity, okay, or the amount of the total phosphatase activity. In physiology, sometimes the important point is the range between the two activities, okay, phosphatase and kinase. Obviously, if the kinase activity is higher compared to phosphatase, the muscle is contract. Is the the kinase activity is higher than the phosphatase. Okay, if the phosphatase is higher, the muscle is released. Okay, it's always the range of the two components. Okay. This is the resume slide, okay? It's the same slide as usual. But the important point that I would like to share with you is this one, okay? Because the activity of the phosphatase alter myosin response to calcium. If the phosphatase activity for some reason is higher, even if the calcium inside of the cell is higher, the muscle cannot be contracted because the phosphatase activity is higher. Okay? Do you remember why? If for some reason I'm able to increase the phosphatase activity, even a high amount of calcium inside do not produce contraction. Why? Because the phosphatase activity is higher. Okay. So for this reason, the relaxation of muscle is due to a multi-step process. When the calcium ion decrease for the activity of different pumps and transporter, the calcium calmodulin complex disappear and the kinase become inactive. But at the same time, as the myosin light chain kinase activity become less active, the myosin light chain phosphatase, the phosphorylate, the myosin, and the myosin ATPase activity decrease and the muscle relax. Okay, it's a multiple step process. The decrease of calcium, but also the high activity of phosphatase.
Let me say, like, because like the phosphatase or the kinase has like a direct relation with the HPase, and so it's controlled either it's relaxed or
exactly. Exactly. This is the point. You can increase the calcium, but if the phosphatase activity, the MLCP activity is higher, even if the concentration of calcium is higher, the phosphatase activity desensitize the myosin. So the contraction cannot take place. Yeah. And physiologically, the phosphatase activity is always present. Okay, so the real activity of the phosphatase is really important because you can increase the kinase activity by the calcium concentration inside, but the phosphatase, the lower level of phosphatase activity is always higher in the cells. So the activity of the increase of calcium is important, but the most important role is the phosphatase activity because it desensitizes the myosin and is always present in the cells. Also because it's the ratio, as I told you before, is the ratio of MLCK and MLCP that really control the state of activation of a smooth muscle. For this reason, because if the ratio, if the MLCP is high, always higher, if you increase the calcium, you can increase the MLCK, but you have to increase a lot of calcium in order the ratio to be bigger.
Yeah. It's like a threshold.
Exactly. And what else? Okay. As I told you before, the membrane potential in small muscle are quite different, okay? Because in panel A and panel B, we have different type of potential, of action potential. In the panel A, we have a slow wave potentials that fire action potential when they reach the threshold. The idea is always the same. If this is the membrane potential of a smooth muscle cell, only when the smooth muscle cell reach the threshold, reach the minus 55 millivolts that we have analyzed for neurons, only in this case the muscle fires action potential. And then if the muscle fires action potential, the muscle can contract because calcium moves inside of the cell during the action potential. Muscle cells can be contracted when the membrane potential moves from a resting potential and reach the threshold. In this case, the threshold generates an action potential, calcium inside of the cell increase, and then the muscle can be contracted. These, in the panel B, are pacemaker potential. Potential in the heart, the cell, that we will analyze tomorrow or the next time, okay? Which is the difference between pacemaker potential and slow wave potential? That in this case, in the pacemaker potential, always the membrane reach a threshold. In the slow wave potential, the membrane can depolarize and repolarize, depolarize and repolarize without giving rise to an action potential. The action potential can be fired only when it reaches the threshold. In the pacemaker potential, we always reach the threshold. In the smooth muscle cells, we reach the thresholds only sometimes. The contraction of muscle cells, smooth muscle cells, caused by electrical signals is named electromechanical coupling. Electromechanical coupling. While a contraction caused by chemical signals is called pharmacomechanical coupling. Pharmacomechanical coupling. But remember, chemical may also, like in the panel C here, chemical may also relax the smooth muscle tension without a changing in membrane potential. This is a pharmacomechanical use of a chemical signal, this one, and when this chemical substance is added to the smooth muscle cell, the smooth muscle cell decrease the tension. Now, what do you think? How is possible to decrease the tension of a muscle? You will study in pharmacology compounds, drugs that can relax the muscle, okay? And their activity is directly on the muscles. How can you relax the muscle from the things that we have analyzed today? How is possible to relax a muscle? For example.
Dephosphorylation.
Exactly. Phosphorylation. We will see activity of some molecules that can increase the activity of the MLCP. So the muscle tension decrease. So Now, the calcium, the point that the calcium initiates the smooth muscle contraction is clear. And the contraction is caused by electrical signaling, and we call it electromechanical coupling, or the contraction is caused by chemical signaling in pharmacomechanical coupling. We know that the calcium initiates the smooth muscle contraction. We know that we can couple the electrical signal in electromechanical coupling of the muscle, but also we know that there are chemical signaling that can produce a pharmacomechanical coupling and the muscle contraction, which will be an electromechanical mechanism that produces, assuming a pure electrical signaling that produce an electromechanical coupling of the smooth muscle cell that produce the contraction. This one. This one. Since calcium ions always initiate the smooth muscle contraction, every voltage gated calcium channel that is present on top of this is, I put here the T-tubule, but we are not in the case of the T-tubule, okay? Because we are in the smooth muscle cell. But in any case, think on a voltage-gated calcium channel on top of the smooth muscle cell. An action potential that arrives here depolarize the cell. And if a calcium channel is present here, calcium moves inside of the cell and activate the contraction from an electrical point of view. Okay.
This one is like the skeleton one.
Exactly, but without the T-tubules. Okay, this is a mistake. Maybe let's think in a voltage-gated calcium channel. Okay, here. And in any case, this is the first type of voltage-gated activation of the muscle contraction. But we have a different source of calcium in the smooth muscle cell that is completely different from the skeletal muscle. But we will analyze it Friday because it's already 11 o'clock. So see you tomorrow. No, see you on Friday. And that's it. Guys, see you on Friday at home.
Nossa, demais, professor.